Hi science killers and welcome back. In today's video lecture we are going to discuss about the blood, and we are going to cover the composition of the blood and the hematocrit value. Hematocrit means volume of red blood cells expressed in percentage, that is called as hematocrit value, and we also name that as peak cell volume or erythrocyte volume fraction. Okay, in order to find the erythrocyte volume fraction or hematocrit value, let's suppose we collect 1 microliter or 1 millimeter cube of blood from a patient in a test tube, and in this case the test tube is called as hematocrit tube or the Winthrop tube. So we collect 1 microliter of the blood in the test tube and we add anticoagulant heparin. We also name that as ethylene diamine tetraacetic acid. So after loading the blood with EDTA in the test tube, we will centrifuge the test tube at a higher speed. So after the spinning of the test tube, the blood collected here in the test tube will be separated out into 3 distinct layers. One layer here at the bottom, and second layer here at the middle, and one last layer here at the top. Okay, the most dense layer of the blood will be packed at the bottom of the test tube, and that is called as packed cell volume, and this layer is of red blood cells. So this layer is of RBCs, red blood cells, which we also call them as erythrocytes. Okay, the second layer here at the middle is called as buffy coat, as its color is white. That's why we call it as white buffy coat. And the third, less dense layer of the blood here at the top is the layer of plasma. So this third layer over here is the layer of plasma. Let's first of all start with the red blood cells or RBCs, and we also name them as erythrocytes. The composition of the red blood cells, according to here, this test tube, that is 1 microliter, its composition is around possibly 0.45 microliter. So in order to find the percentage, we will divide it with the total number, that is 1 microliter, and we will multiply that with 100. So its percentage will be 45%. So this is what we say, that blood is composed of 45% of red blood cells. So this 45% is actually the hematocrit value, which we also name as peak cell volume. The normal value of the red blood cells is around 5 to 6 million per microliter. So here we get 2 conditions. In the case, let's suppose if there is a patient and if he has less than 45% of red blood cells, we will call this condition as anemia. And if in the case he has got more than 45% of red blood cells count, so we'll call this condition as polycythemia. So this was all about the red blood cells or erythrocytes. Next layer here at the middle, that is white buffy coat, is composed of two things. So this layer is actually formed by the aggregation of WBCs and platelets. So here we have got number one, white blood cells, WBCs, or leukocytes, and another thing over here, that is platelets, which we also name as thrombocytes. Normal value of WBCs is around 4,800 to 11,000 per microliter, and normal value of platelets is around 150,000 to 450,000 per microliter. So here also we have got two conditions. In the case platelets, they are less than, if the platelets are less than 150,000 per microliter, we will call this condition as thrombocytopenia. And in the case if he has got platelets more than 450,000, so this condition is called as thrombocytosis. So this is called as thrombocytosis. Similarly is the case with the WBCs. If in the case he has got less than 4,800 white blood cells per microliter, so this condition is called as leukopenia. And if he has got more than 11,000 per microliter of the WBCs, that condition is called as leukocytosis. So here we have covered the two things, the RBCs and the white buffy coat. RBCs, platelets and WBCs, these are actually the formed elements of the blood. Okay, the next layer here at the top, that is the less dense part of the blood, that is plasma. The composition of the plasma in 1 microliter is around 0.55 microliter. To find the percentage, we will divide it with the total number, that is 1 microliter, and multiply it with 100. The answer we will get here is 55%. So plasma in the blood is around 55%. So blood is composed of 55% plasma and 45% red blood cells or erythrocytes. In plasma we have a couple of constituents and elements. Plasma is composed of 90 to 93% water, and we have 8% proteins. We call them as plasma proteins. Like out of these proteins we have 60% albumin transport protein. We also have in plasma globulin proteins, like alpha and beta globulins and gamma globulins. Alpha and beta globulins are actually the transport proteins, and they transport the metals. One of the best example is transferrin. There's also alpha and beta globulin. So this transferrin actually transports iron. Okay, gamma globulins are also called as immunoglobulins, and they are called as antibodies, and they function in the defense system of our body then. Okay, next we have another protein that is not present in the serum, that is called as fibrinogen. Albumin and globulins, they are present in the serum. That's why they are called as serum albumin and serum globulin. while fibrinogen is absent in the serum, but yes, fibrinogen is still present in the plasma. So this is absent in the serum. Fibrinogen functions in the coagulation cascade. So fibrinogen is actually playing a very, very important role in the hemostasis, in the blood stopping them. Okay, next we have gases in the plasma like oxygen, carbon dioxide, nitrous oxide. We also have nutrients in the plasma like glucose, fats, amino acids, essential amino acids and non-essential amino acids. They are also present in the plasma. We also have metabolic wastes in the plasma like lactic acid, uric acid, urea, creatinine. They are also nitrogenous wastes, and they are present in the plasma. We also have enzymes. And hormones. They are also present in the plasma. So this was all about the composition of the blood, and it was all about the hematocrit value. Here one thing at the last I would like to mention, and that is serum. This is very, very important to differentiate between plasma and serum. In order to understand the serum, let's suppose we have taken blood and we have collected the blood in a container. Once you collect the blood in a container, what will happen? That blood formation will take place. How? Because the fibrinogen present in the blood will be converted. This fibrinogen present in the blood will be converted into fibrin. So this fibrin mesh, network like mesh is there, that is actually formed by the fibrin. This fibrin will entrap the red blood cells. So these red blood cells will not be able to move forward. So as a result, what will happen, that a such kind of clot will be formed. So this is called as blood clot. So this is called as blood clot. So after 45 minutes, once the clot has been formed, after possibly 45 minutes, serum oozes out. So serum is nothing but same plasma except for, or in the absence of, fibrinogen. So we'll see that yes, serum is actually equal to the volume of same 55% plasma is here, same 55% serum will be there. So serum is equal in the volume to the plasma if you minus fibrinogen from plasma. Say this is a very simplest formula to differentiate between plasma and serum, that serum and plasma both are same. Plasma has all proteins and serum has all proteins except for fibrinogen. So it means in serum fibrinogen protein is absent because fibrinogen has been already converted into the fibrin. So fibrinogen is soluble part and fibrin is the insoluble part. So guys, here you have a very strong information about the composition of the blood. That yes, blood is composed of plasma and the formed elements that are actually platelets, WBCs and RBCs. So I hope you did enjoy and I hope it helped you a lot. So if you did, please press the like button and the bell icon, and please subscribe to my YouTube channel, like the video and share the video. I thank you all.